Hello friends, dear students and children. Today I am going to discuss about biomolecule, a very important topic for your CBSE science stream, NEET exam, GATE exam, means all different type of competitive exam. So first of all, I am going to tell a basic concept just to revise topic, that is biomolecules. Okay, so just I'm going to discuss the different type of bond, chemical bond present in biomolecule. First, the glycosidic bond, a bond between two units of glucose in carbohydrate, and the bond between nitrogenous base and ribose and deoxyribose of RNA and DNA. So in that case, mainly in carbohydrate and in DNA or RNA, this glycosidic bond present. So there are two type of glycosidic bond. One is O-Glycosidic bond, that is present between two unit of glucose in oligosaccharides and polysaccharides. But if you observe the single stranded DNA or single stranded RNA, in that case N-Glycosidic bond present between nitrogenous base and ribose or deoxyribose sugar. Next type of bond present in biomolecule is peptide bond. So a peptide bond generally present between two unit of amino acids in protein and polypeptide chain. Next type of bonding is phosphodiester bond. That type of bond present between two units of nucleotides in nucleic acid, mainly in DNA and RNA. Next question may be asked, example of disaccharides are. So these are lactose, it is made up of glucose and galactose. Maltose, it is made up of glucose and glucose. And sucrose, it is made up of glucose and fructose. So you must remember example of disaccharides. Example of non-reducing sugars are sucrose and all polysaccharides, that is starch and glycogen. Starch is present in plant and glycogen is present in animal muscles. Next, example of reducing sugar are all monosaccharides. Monosaccharide means simplest sugar. It may be glyceraldehyde, it may be uh, pentose, ribose, or glucose, galactose, and disaccharide, that is lactose and maltose. So these all are reducing sugar because hydroxy group present at carbon number 1 is free and it can react with any reducing agent or any chemical reagents. So it will give color reactions. Next is, what is carbohydrate? You know, you can recognize carbohydrate by presence of aldehyde group and then ketone groups and then many hydroxy group. So carbohydrate is aldehydic or ketonic form of polyhydroxy alcohol. Either aldehyde will present or ketone will present. Next, carbohydrate is tested through iodine for a starch. You can test the starch by iodine solutions. It will give blue color or blackish color. Thalins and Vandyck solution is test solution for testing for monosaccharides, mainly glucose. Next, what is protein? Protein is macromolecular, linear polymer, high molecular, organic, not organic, inorganic, organic weight compound joined together by peptide bond. Mainly peptide bond make a polypeptide. Okay, so this is joined together by peptide. Protein and amino acid is tested through which test? That is ninhydrin. It will gives blue colors. Okay. Next, essential amino acid are which are not synthesized in our body and taken through your diet, through your food. So you must remember example of essential amino acids generally frequently asked in all the competitive exam, in all your CBSE exam, and you remember like that. Treat very ill MP at hospital. So I have darkened the letters that a single letter symbol of different amino acid, that is tryptophan, T for tryptophan, T for threonine, V for valence, I for isoleucine, L for leucine, and L for lysine, M for methionine, P for phenylalanine, A for arginine, and H for histidine. So there are 10 essential amino acids. Rest 10 are non-essential amino acids. So total 20 standard amino acid, 20 standard different type of amino acid present in your body. So if you see the different example of the amino acids, standard amino acid, non-standard amino acid. So these are the very important for your competitive point of view. So you must revise carbohydrate and proteins in the same way for complete the question in all the competitive exam. Thank you.